Welcome back to Anatomy and Physiology on Catalyst University. My name is Kevin Tokoph. Please make sure to like this video and subscribe to my channel for future videos and notifications. In this video, we're going to discuss something in bacteria called the lac operon. This is a very common subject to discuss in a biochemistry course or cell biology. You might even see it in some other courses. So what is the lac operon? Well, first of all, the lac operon is something that we only see in bacteria. This is not something that we see in eukaryotes. So this is specific to bacteria. And the purpose of the lac operon is to regulate lactose metabolism. Lactose is a disaccharide. It's actually a disaccharide of glucose and galactose, and bacteria can metabolize lactose. And so if there is no lactose available, then we wouldn't need any enzymes or proteins that are involved in lactose metabolism, because if there's no lactose, we wouldn't need them. And it would make sense not to make those proteins and enzymes. And if there is lactose present, in some cases the bacteria might want to utilize that lactose for energy. And so the bacteria would have to have enzymes and proteins necessary for lactose handling and metabolism. And these are actually the enzymes right here in proteins. We'll discuss these more in a few minutes, but they're usually called LAC-Z, LAC-Y, and LAC-A. But there's one key thing to remember here with these bacteria. Yes, they can metabolize lactose. However, glucose is the preferred energy source. So that being said, even if there is lactose, if there's glucose present, they will preferentially want to metabolize the glucose, and they really won't deal much with the lactose. They really want the glucose really because it's just easier, and glucose metabolism is more universal. All right. Now, in terms of these enzymes, LAC-Z, LAC-Y, and LAC-A, these are actually the gene names, but they actually encode specific proteins. So the first one we'll talk about is LAC-Y. The LAC-Y gene encodes a protein called lactose permease. This is just a membrane protein that facilitates the transport of lactose from outside the cell into the cell. Okay. So in order for the bacteria, which is this cell right here, in order for the bacterium to actually metabolize lactose, it would obviously have to get the lactose into the cell first. That's the job of LAC-Y, or our lactose permease. Sometimes you might hear it referred to as galactoside permease, because lactose is a type of galactoside, and there are other galactosides that are similar to lactose, but this protein will get all of those into the cell. The second protein is actually an enzyme. It's LAC-Z, and it's called beta-galactosidase. It pretty much is a similar enzyme to what humans have, the enzyme lactase, but generally from the perspective of the lac operon, it'll be called beta-galactosidase. This is a hydrolytic enzyme, and what it does here is it splits lactose, the disaccharide, into its two constituent monosaccharides, glucose and galactose. And then both of these, especially the glucose, can be utilized for energy production via glycolysis or whatever pathway that bacterium uses. All right? And that's the major reaction of beta-galactosidase. However, what's really important to understand is that the beta-galactosidase also has a side reaction that occurs alongside the major reaction. Now, the major reaction might account for 99% of the reactions, but there's a small 1% side reactions where lactose is converted to an isomer called allolactose. And we're going to see allolactose will actually have some functions in terms of the regulation of the lac operon. And even though there's only about 1% of these side reactions actually occurring, allolactose is important in that regulation. Now, the last enzyme is going to be LAC-A. Now, typically when you look up LAC-A, you'll see that the function is, for the most part, unknown. They're not really solidified on exactly what it's for, but the most prominent hypothesis is that it's involved in the excretion of toxic galactosides. So some galactosides that might be taken into the cell might interfere with certain processes, they might be toxic, and so the cell's going to want to get rid of those. So here's a toxic galactoside. LAC-A encodes the protein galactoside transacetylase. This is an enzyme that attaches the acetyl group from acetyl-CoA onto the galactoside, and so you end up getting an acetylated toxic galactoside, which can then be moved out of the cell. So this enzyme is involved in acetylating toxic galactosides, and then those can then be moved out of the cell so that they don't interfere with metabolism and kill the bacterium. Okay. Again, this function is not exactly well understood, we're not really going to focus on it. But the major thing to understand is that whenever you get one of these enzymes or proteins, you get all of them. Okay, so they're all important to some extent. Now, whether or not we get these proteins, whether or not we get them expressed, depends on if we have glucose, if we have lactose, if we have both, or neither. So let's look at first a case where we have lactose, thus it's lactose positive, but we do not have glucose. And we have some major players here. So this protein right here, this is an enzyme. This is RNA polymerase. Now, of course, if we wanted to get these 3 genes transcribed in the operon, we would, of course, have to have RNA polymerase attached to the promoter, which is LAC-P. This is the promoter of the LAC operon. And then you'd, of course, get transcription. Okay? But the RNA polymerase has to attach. 2 other players. First of all, we have this CAP. This is cyclic AMP binding protein, or just CAP. And there is a region of the operon where CAP will bind. Okay, so here's CAP. And then over here, we have the LAC repressor. Okay, the LAC repressor, if it's activated, will bind to this region called the operator, the O, and it will prevent this RNA polymerase from binding. Okay, so there's 3 players here. Let's talk about it. All right, so in our situation here, we only have lactose available. There is no glucose, and that's a key. We have no glucose, only lactose. Well, the bacteria needs energy. It needs to metabolize something, so if there's no glucose, it can't use that. It will have to use lactose. And so therefore, we would expect to get transcription of all 3 of these enzymes, right, and expression. And so how does that happen? So first of all, let's look at lactose. So there's always going to be some level of these enzymes right here, the beta-galactosidase, always some level, okay, even if it's very small. And so you're going to get some of this lactose that's converted to allolactose, all right? Now what allolactose can do, as you see right here, is it can bind to the LAC repressor, okay? When allolactose binds to the LAC repressor, that inhibits the LAC repressor. Now normally, if there were no allolactose, the LAC repressor would come up here, bind to the operator, and prevent RNA polymerase from binding, and therefore you'd get no transcription. However, in this case, what's happening is this allolactose, this baseline allolactose that's being generated, kind of turns the LAC repressor off and prevents it from binding to the operator, and therefore that allows RNA polymerase to bind. Okay? Now, there's only going to be a little bit of allolactose, and that's all it takes. But whenever there's allolactose, that's indicative of the fact that there is lactose. Okay? So when there's lactose, there's also allolactose, and allolactose binds to the LAC repressor and turns it off, prevents it from binding to the operator. All right? That's half of the story. The more important half is really the fact that there's no glucose. So here's something I want you to remember. If there were high glucose, lots of glucose present, there would be no cyclic AMP. Okay, this is just a molecule, cyclic AMP. In contrast, if there's very little glucose, we'd have very high levels of cyclic AMP. So here we have no glucose, so we would expect there to be a lot of cyclic AMP. Well, this protein right here, CAP, it is a cyclic AMP binding protein. So the fact that we have no glucose means cyclic AMP is high, and then this cyclic AMP comes over and binds to CAP, all right, this protein. And when cyclic AMP binds to CAP, then CAP binds to the CAP binding region. And when CAP is bound to this CAP binding region, that facilitates RNA polymerase to bind to the promoter. Okay? So really, you really have to have 2 things here present. You have to have CAP bound to its binding region on the DNA, and you also have to have the LAC repressor not bound. Okay? Now, if there was a lot of glucose present, we wouldn't have this cyclic AMP, and then this CAP binding protein would not be bound here, and then RNA polymerase would not be bound. Okay? But the fact is, we have cyclic AMP, lots of it. That binds to CAP, that activates CAP, allows it to bind to the CAP binding region, which then allows this RNA polymerase to come over here and bind to the promoter. And in conjunction with the inhibition of the LAC repressor, we get transcription of all 3 of these genes. Okay? So the net effect when we have lactose present but no glucose is we get transcription of all 3 of these genes. We get LAC-Z, LAC-Y, and LAC-A. All right, and therefore we can now get the lactose into the cell, and then through this enzyme beta-galactosidase we can then metabolize it into glucose and galactose for energy production. Okay. Now we're going to see a very different situation when we add in glucose. So here we have a situation where we have both lactose and glucose. And you might say, well, there's two nutrients, so the bacteria will metabolize them equally. That is not the case. Okay? Remember that for these bacteria, glucose is the preferred energy source. So despite the fact that there is lactose present, these bacteria are going to focus all their effort and attention on metabolizing the glucose. They're sort of going to leave the lactose alone. Okay? There will be, as we'll see, negligible transcription of these three genes, but how does that occur? Right? Well, again, we still have some allolactose. Okay? There's going to be a baseline level of allolactose. You don't need much of it, okay? Because there's always going to be a little bit of that beta-galactosidase activity. And so lactose is going to be converted to allolactose, and allolactose is going to inhibit the lac repressor. Okay? Remember, when allolactose binds to the lac repressor, that lac repressor can't come over here and bind to the operator. And if that were to occur, okay, that might actually prevent RNA polymerase from binding. Okay? But again, this allolactose is keeping this lac repressor from binding to the operator. So that brings up the question, why don't we get transcription of these genes? It's because now we have no cyclic AMP. It turns out that for getting transcription of these three genes, this cap protein is much more important than the lac repressor. Okay? In order to get transcription of these three genes, you really have to have this cap protein bound there. Okay? But it will only bind there if we have plenty of cyclic AMP present. But remember, if we have high glucose, we have no cyclic AMP. So elevated glucose levels mean no cyclic AMP, and therefore no activity of cap. So cap cannot bind here. Okay? It cannot bind to the cap binding region. Therefore, the binding of RNA polymerase is going to be very, very weak. It can still bind a very, very small amount because the lac repressor is inhibited. But overall, because cap cannot bind here, the RNA polymerase binding to the promoter is going to be negligible. And so that being said, we're going to have negligible transcription of these three genes. There's going to be a very, very, very small amount of it. But we don't really need a lot of it, right? Because we've got plenty of glucose present here. And the bacteria will preferentially metabolize that glucose. And so the key thing to remember about the function of the lac operon, the cap is much more important for regulating the transcription. And so if there's no cyclic AMP due to high glucose levels, cap is going to be inhibited and you won't get that binding and negligible transcription, which is okay because we've got plenty of glucose. Now, the last case we're going to look at is what happens when there's only glucose available. So there's no lactose and there's plenty of glucose. Well, when there's no lactose, there's no allolactose. Okay, remember that in order to get that allolactose, you have to have some degree of lactose, right? Because that baseline beta-galactosidase level is going to have that side reaction to convert lactose into allolactose. Then the allolactose will in turn inhibit the lac repressor and prevent it from binding to the operator. However, if there's no lactose, there is no allolactose. And therefore, this lac repressor is no longer inhibited. It can come over and then bind to that operator. Okay. And that operator being bound to is partial inhibition of the lac operon as a whole. But again, the cap protein is much more important. So we have high glucose. That means no cyclic AMP, and therefore this cap protein is going to remain inhibited. This cap protein can't come over here and bind to the cap binding region. And so if you look at no cap, meaning no activation of the operon, and you have full inhibition via the lac repressor, you have absolutely no binding of the RNA polymerase and absolutely no transcription. If you looked over here in the previous example where we had both nutrients, we had inhibition of the lac repressor. So actually neither of these were actually bound. And so there was just a little bit of transcription of these three genes. In this case, the LAC repressor is actually activated. And so in conjunction with the fact that the cap binding protein is inhibited, you get absolutely no binding of RNA polymerase, none. And so none of these 3 genes will get expressed, and so you'll have none of these 3 proteins, which is okay, because even though we have no lactose, we've got plenty of glucose, which will then be converted to energy through various pathways. And I mentioned a few of them here: glycolysis, the Entner-Duderoff pathway in bacteria, or hexose monophosphate shunt. But in any case, this is how the LAC operon works. Okay? If you want transcription of these genes, you have to have cap bound to the cap binding region. That's the activating region. And you really can't have this operator bound to by the LAC repressor, so the LAC repressor ought to be inhibited. And when those 2 things are true, you have maximal binding of this RNA polymerase, maximal transcription, and you get these 3 enzymes or proteins transcribed and expressed, and then you can metabolize lactose. But the key is that can only be when lactose is present and glucose is absent. As long as there's glucose, the cell will really not be transcribing these either negligibly or at all, and that cell will be preferentially metabolizing the glucose. Okay? So hopefully this made sense to you, and you have a little bit of a better understanding of the LAC operon. Please make sure to like this video and subscribe to my channel for future videos and notifications. Thank you very much.